what's up everybody? It's Kirsten and we have some hot balance topics for you today. Today we'll be discussing the five sense organs, the otolithic organs, and the vestibular nerve pathway, and the horizontal semicircular canal vestibulo-ocular reflex. So we will start off with the semicircular canals. So there are three semicircular canals that are organized in 90-degree angles, and these canals detect angular head movement in three different directions. So the horizontal semicircular canal detects movement in the yaw plane, which I will demonstrate now as this movement. And the anterior semicircular canal detects movement in the roll plane, which is this movement. Don't know if you heard my neck crack there. And the posterior semicircular canal detects movement in the pitch plane, which is this movement here. So these canals are formed in the bony labyrinth of the vestibule, and each canal consists of a bump/hump-like structure, also known as the ampulla. Housed within the crista ampullaris, you will find type 1 and type 2 hair cells. Each of these hair cells consist of stereocilia and kinocilia, which extend into a gelatinous mass called the cupula. The kinocilium is surrounded by many stereocilia, which are deflected into different directions by the flow of endolymph. So in this image here, you can note that the kinocilia is the much larger hair cell, while the stereocilia are the much more shorter hair cells here. So these hair cells are deflected in different directions by the flow of endolymph. In the anterior and posterior canals, the ampulofugal flow means the endolymph movement deflects the stereocilia towards the kinocilia, causing excitation. So demonstrated here, as the endolymph flows through the canal, the stereocilia are deflected towards the kinocilia. In the horizontal canal, ampulopedal flow means the endolymph movement deflects the stereocilia towards the kinocilia. Excitation causes an increase in the firing rate of the nerve. However, ampulopedal flow in the anterior and posterior canals cause inhibition, which is demonstrated here in this image. As the endolymph flows through the canal, the stereocilia are deflected away from the kinocilia. And ampulofugal flow in the horizontal canal causes inhibition. Inhibition causes a decrease in the firing rate of the nerve. So for example, if Ricky turns his head to the left while in the yaw head position, the left horizontal semicircular canal will be excited and ampulopedal flow, while the right horizontal semicircular canal is inhibited with ampulofugal flow. So, as you can see, this would be the left horizontal semicircular canal, which is excited when turned to the left, while the right semicircular canal is inhibited in that head turn, so that the brain is able to detect which plane or direction that the head is turned. So now that we've discussed the semicircular canals, we'll now move on to the otolithic organs, also known as the utricle and saccule. They respond to linear acceleration and changes to head position. Both of these organs have macular structures inside of them. The macular organ within the saccule houses the sensory organs for the vertical plane, while the macular organ within the utricle houses the sensory organs for the horizontal plane. Superior to the macular sensory epithelium, noted here, you will find the hair cells, which project into a gelatinous mass known as the otolithic membrane. On top of the otolithic membrane, you will find ear rocks or otoconia made of calcium carbonate, which when displaced assist the otolithic membrane in movement of hair cells. In the saccule, up here, you will note that the kinocilia are arranged in opposing positions and are separated by the striola, this dashed line here in the middle. And if you forgot, our kinocilia are the much larger hair cells, and our stereocilia are the much shorter hair cells. Deflection of the otoconia in one direction causes excitation and inhibition within the macular organ. In the utricle, over here, the kinocilia are oriented towards the striola here in the middle. Deflection of the otoconia causes excitation and inhibition within the macular organ as well. So when linear acceleration occurs, hair cells on one side of the striola are excited, while the hair cells on the other side of the striola are inhibited. Hi there. So now we'll be discussing the vestibular nerve pathways, the afferent nerve pathway that is, and the sensory organs of the vestibular system send their afferent nerve pathways up to what is called Scarpa's ganglia. From Scarpa's ganglia, the nerve travels through the inner auditory canal to the cerebellar pontine angle, where the vestibular nerve synapses with the vestibular nuclei located in the brain. At the cerebellar pontine angle, the vestibular nerve bifurcates into two different branches. The superior branch innervates the utricle horizontal semicircular canal and the anterior semicircular canal, and part of the saccule. The superior branch travels through the CPA, or cerebellar pontine angle, to synapse with the superior and medial vestibular nuclei. The inferior vestibular nerve innervates the saccule and posterior semicircular canal and synapses with the medial, lateral, and inferior vestibular nuclei. Alright, thanks for hanging in there, guys. So now we'll start discussing the horizontal semicircular canal vestibulo-ocular reflex, also known as VOR. The VOR main purpose is to help maintain clear vision while the head is in motion. This reflex consists of three innervation arcs. Reflex arc 1, noted here by the solid black line, sends a signal from the horizontal semicircular canal to the medial vestibular nucleus. This signal then travels across the midline, noted here by the dashed line, to the abducens nerve. The abducens nerve then sends that signal to the lateral rectus on the contralateral side and abducts the eye. So now this results in eye movement opposite of the head movement. So reflex arc 2 sends a contralateral signal, and reflex arc 2 is noted here by the dashed line. It sends a contralateral signal from the abducens nerve over to the ipsilateral oculomotor nucleus. The oculomotor nucleus then sends a signal down to the ipsilateral medial rectus, and that then adducts the eye. Lastly, reflex arc 3, noted by the blue line here, sends a signal from the horizontal semicircular canal to the superior vestibular nucleus, which innervates the oculomotor nucleus, which then sends a signal down to the ipsilateral medial rectus, which results in the eye being in an adducted position. Alright guys, thank you for watching my balance video. Hope you enjoyed it. Make sure to like, comment, and subscribe down below. And I'll see you next time.